Pick up your morning coffee and breathe in. You just did something effortless, something you have done your whole life. You also just ran a physics experiment that the best scientists in the world cannot fully explain. Nobody knows, at the deepest level, how your nose turned a few drifting molecules into the experience of coffee. This is the second lesson in my quantum biology series. In the first, I tried to separate what is settled from what is contested from what is merely hoped for, because the honest version of this field is far more interesting than the headline version. Smell is the case where that honesty matters most. It is the one place where a bold quantum idea was proposed, made a sharp prediction, and was put on trial. What survives is smaller, stranger, and still open. Start with the size of the problem. You have about 400 kinds of smell receptors in your nose. That is the largest family of genes you carry, but it is small next to the number of odors you can tell apart. So the nose does not use one receptor per smell, the way a lock uses one key. It uses combinations. One odorant lights up a pattern across many receptors, and one receptor answers to many odorants. Your brain reads the pattern, not the piece. That much is Nobel Prize winning and settled. The harder question is what a single receptor is actually measuring when a molecule lands on it. The mainstream answer is shape. A receptor is a pocket, an odorant is a key, and recognition happens when the molecule fits. It has real evidence behind it. Carbon is the cleanest case. It comes in two mirror image forms, identical except for handedness. One smells of spearmint, the other of caraway, and your receptors, which are themselves handed, can tell them apart exactly as shape predicts. But shape is not the whole story. Molecules of very different shape can smell alike, and almost anything carrying a sulfur-hydrogen group smells of rotten eggs, no matter its overall form. Something about certain chemical features seems to carry a smell of its own. In the 1990s, the biophysicist Luca Turin sharpened a radical alternative, that the nose does not only feel a molecule's shape, but listens to it. Every molecule vibrates, its bonds stretching and bending at frequencies unique to its chemistry, a kind of infrared signature. Turing argued the receptor reads that signature through a quantum process called inelastic electron tunneling. In plain terms, an electron slips across a gap it should not be able to cross, and it can only complete the jump if the molecule sitting in the gap is vibrating at just the right frequency to absorb the leftover energy. We already build machines that work exactly this way. For years, this was dismissed as physically loose talk until 2007, when a group of physicists ran the numbers and showed the mechanism is not forbidden by physics at all. That did not prove the nose works this way. It did something more useful. It turned a poetic idea into a model sharp enough to test and to disregard. Here is the beautiful part. If the nose reads vibration, then two molecules with the same shape but different vibrations should smell different. Take a molecule and swap some of its hydrogen atoms for deuterium, hydrogen's heavier twin. The shape barely changes, but the vibration drops by about half. Shape theory says they should smell identical. Vibration theory says they should smell different. Total disagreement. And it was tested at the receptor itself. In 2015, a team took the human and mouse receptors that respond to musk, exposed them to ordinary and deuterated versions, and asked whether the receptor could tell them apart. It could not. The strong form of the theory, the version that says smell is vibration sensing, had made its prediction at the level that counts, and lost. A lesser story would stop there, with a tidy verdict. Shape wins, vibration loses. The real situation is more honest. What died is the strong claim that receptors read vibration instead of shape. What survives is a hybrid, sometimes called the swipe card model, A card has to be the right shape to enter the reader, and only then is the stripe read. Perhaps a molecule must first fit the pocket, and only then does a quantum step help pull the trigger. That version is much harder to test. And the frontier is still live. As recently as this year, physicists went on modeling odorants as weak quantum conductors. Notice how many rooms of the university you just walked through. The receptors and the combinatorial code are neuroscience. The vibrations and the deuterium trick are chemistry. The tunneling electron is quantum physics. And underneath all of it sits evolution, because smell is the oldest sense we have. Long before anything had eyes or ears, a single bacterium could read a chemical gradient and swim toward food. Whatever mechanism our noses use has had something like 3 billion years of evolutionary search behind it. That is what makes the question serious. The honest answer today is that a living cell is warm, wet, and noisy, an environment that destroys delicate quantum states almost instantly. So any claim that biology uses one has to clear a very high bar. Photosynthesis and magnetoreception clear it. Olfaction, so far, does not. So here is my question for you. When a bold scientific idea gets tested and mostly fails, what should we do with it? Do we throw it out entirely? Keep the humble piece that survived? Or hold it open until better tools arrive? Tell me in the comments. One more thing before we close. If this way of thinking across fields is something you would like to build in yourself, the Renaissance Hub Idea Ranker is a free place to start. The link is in the description. We do not yet know how you smelled your coffee this morning. Not fully. And that unfinished sentence is not a gap to be embarrassed about. It is an invitation. The wonder survives the scrutiny. It usually does. For more, visit santiagolab.org. Remember to be curious as children and let nature surprise you. If you liked the video, please comment, subscribe, share it, and give us a like. Stay Renaissance.